Okay, is presentation visible? Hello.
Visible, sir. Visible, sir. Visible.
Okay, so today's topic is pentose phosphate pathway. So, as name suggests, it is related to pentose sugar. Okay, so under carbohydrate we have pentose, hexose, triose, likewise. So this is related to carbohydrate pathway, and this is a pentose. Okay, pentose phosphate means five carbon sugar is there, and there is a phosphate group attached with this pentose sugar. So this is why it is called as a pentose phosphate pathway. So under chemical reaction, there is a concept of standard free energy. So delta G, the free energy of reaction under standard condition is defined as concentration of reactants and product at 1 molar concentration and temperature at 25 degrees Celsius and acidity at the 7. So if this standard condition is there, means reactant and product concentration is 1 molar each, and temperature is 25 degrees Celsius, and acidity means pH is the 7. Under this condition, for to delta G, we call it as a standard free energy change. Okay, so the last lecture we are shown some delta G. So here is the explanation for this one. So pentose phosphate pathway. Coming to our point, pentose phosphate pathway. It is also called as a pentose shunt or hexose monophosphate shunt. Okay, this pathway, pentose phosphate pathway, is also known by the name pentose shunt or the hexose monophosphate shunt. And there is one more name to this one, phosphogluconate pathway. So this phosphogluconate pathway name is also there, and it occurs in the cytosol. Okay, our glycolysis we saw, it also occur in the cytosol, and this pentose phosphate pathway, this also occur in the cytosol. So now coming to this point, so what is happening? One of the fate of glucose 6-phosphate is the pentose pathway. So whenever there is a glucose 6-phosphate present, what can happen to this glucose 6-phosphate? So this is our glycogen. This is our glycogen. If there is an enzyme glycogen phosphorylase, so what happens is that the glycogen, the glucose gets phosphorylated, and we get here glucose 1-phosphate. So from glycogen, one glucose gets removed. We call it as a glucose 1-phosphate, and enzyme involved is a glycogen phosphorylase. So glucose 1-phosphate is there. Now with the help of phosphoglucomutase, this glucose 1-phosphate can get converted into the glucose 6-phosphate. Okay, so basically glucose 1-phosphate and 6-phosphate structurally they are similar atoms, carbon atom, phosphate atom, but at the glucose 1-phosphate it is attached at the first carbon position, and in case of glucose 6-phosphate, the phosphate group is attached at the 6th carbon position. Now glucose 6-phosphate is there. What can happen to glucose 6-phosphate? Under glycolysis we saw this glucose 6-phosphate can undergo the process of glycolysis in the muscle, brain, and it can yield end product as a pyruvate. And we also saw fates of pyruvate. Pyruvate can get converted into lactate or carbon dioxide and water, or pyruvate can get converted into the ethanol. So this is one glycolytic pathway. Next, what can happen with this glucose 6-phosphate? So liver has an enzyme glucose 6-phosphatase. If this enzyme is present, this 6-phosphate group get removed, and we can get glucose from the glucose 6-phosphate. So the blood for it can be used in the blood for by other tissues. So this is basically gluconeogenesis pathway. Next, glucose 6-phosphate is there. This glucose 6-phosphate can undergo this pentose phosphate pathway, and we can get the ribose. Ribose is a 5-carbon sugar. So ribose sugar is a 5-carbon aldose sugar. Okay, so this ribose sugar is made up of 5-carbon. So that is why this is pentose phosphate pathway, and here NADPH is present. Okay, under the glycolysis or Krebs cycle, we saw there is the presence of NADH, but in this case, the pentose phosphate pathway, it produces the NADPH. So this phosphate group is additionally present here. So the NADH and NADPH, some difference is there. Here additional phosphate group is there. So this is a reducing equivalent produced in the pentose phosphate pathway. Okay, so basically our aim is that we get ribose here and the NADH here. Okay, so pentose phosphate pathway is a shunt. The pathway begins with glycolytic intermediates, glucose 6-phosphate. We saw here, okay, this is glycolytic intermediate, glucose getting converted into glucose 6-phosphate, and this intermediate can serve as a substrate for this pentose phosphate pathway. So this is a glycolytic intermediate of the glucose 6-phosphate. It connects with the glycolysis because two of the end products of the pentose pathway are glyceraldehyde 3-phosphate and the fructose 6-phosphate. Okay, so uh, this pathway, pentose phosphate pathway, connect with this glycolysis also, because the end product of this pentose pathway, that is glyceraldehyde 3-phosphate and the fructose 6-phosphate, they can be intermediates for the glycolytic pathways. Okay, it is for this reason that pentose phosphate pathway is also called as a shunt. So that is why it is also called as a shunt. So here, for example, this one is a moderate glucose flow. So this is, for example, glucose is there, only one pathway is there. So this the water is falling only by one way, glucose only. But in case of large glucose flux, water can fall, means glucose can undergo this phase or glucose can undergo this phase. So that is why it is called as shunt. Okay, pentose phosphate pathway. So how this shunt terminology work? So basically glucose is there. Glucose gets converted into the glucose 6-phosphate with the help of enzyme hexokinase. Manganese is also required in this process, and this ATP is getting converted into ADP. Okay, this is first step of the glycolysis, formation of glucose 6-phosphate. Next, glucose 6-phosphate with the help of phosphoglucoisomerase get converted into the fructose 6-phosphate. So this is isomerase reaction, and we are from glucose 6-phosphate we are getting fructose 6-phosphate. Next, with the help of phosphofructokinase, we are getting fructose 1,6-bisphosphate. And during this stage, ATP is incorporated, and here fructose 6-phosphate is getting converted into the fructose 1,6-bisphosphate. And during at the next stage, the fructose 1,6-bisphosphate with the help of aldolase enzyme get cleaves into the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. And we already saw this glyceraldehyde 3-phosphate can get interconverted with the help of the triose phosphate isomerase into the dihydroxyacetone phosphate. So this is basically glucose 2. These steps are the glycolysis steps. So where this so alternative to this glycolysis pathway, there is alternative pathway, we call it as a pentose phosphate pathway. So what happens is that this glucose 6-phosphate is there, it can enter into this pentose phosphate pathway, or these intermediates at the end product can enter as a glucose 6 fructose 6-phosphate, or in this product the reaction pentose phosphate pathway, this glyceraldehyde 3-phosphate can form. So glucose 6-phosphate, fructose 6-phosphate and the glyceraldehyde 3-phosphate, these are the three things which are common to this glycolysis and the pentose phosphate pathway. So what does the pentose phosphate pathway achieve? The pathway yields the reducing potential in form of NADPH. So in this pathway, pentose phosphate pathway, we are getting reducing potential. Okay. So whenever any anabolic reaction is there, means some synthesis of fatty acid is there, or any synthetic reaction is there. So we, for synthesis reaction, we call them as an anabolic. If some substrate is breaking down into smaller parts, we call it as a catabolic reaction. So during anabolic reaction, this reducing potential is needed in the form of NADPH. So in anabolic reaction, the NADPH is used in the anabolic reaction requiring the electrons. So during anabolic reaction, some electrons are required. So if this NADPH is there, it says, okay, I am ready to use the electron. So this is the role of NADPH, which is produced in the pentose phosphate pathway. The pathway yields a ribose 5-phosphate. So ribose 5-phosphate. So already we are familiar with the nucleic acid. There are two types of nucleic acid, like deoxyribonucleic acid and ribonucleic acid. So under nucleotides, whenever DNA is there, there is a deoxyribose sugar is present and RNA ribose sugar is present. So here the ribose is involved in the DNA, RNA, and in the various cofactor like coenzyme A, FAD, S-adenine methionine transferase, or SAM, or NAD plus or NADP plus. In this case also, this ribose 5-phosphate is needed. So basically, aim of the pentose phosphate pathway is that here we are getting some reducing potential in the form of NADPH, and we are getting sugar as a pentose sugar. We call it as a ribose 5-phosphate. So where does this pathway NADPH is required? We say we get this NADPH molecule in the pentose phosphate pathway. So where its role is there? So whenever any synthetic reaction is there, for example, fatty acid biosynthesis reaction is there, in that case you may require NADPH, cholesterol biosynthesis require NADPH, the neurotransporter biosynthesis reaction require NADPH, nucleotide biosynthesis reaction require NADPH, some detoxification reaction like reduction of oxidized glutathione. Whenever glutathione is in oxidized form and we want to make it reduced, in that case also NADPH is required. Cytochrome P450 monooxygenase require this NADPH. So these are the reaction in which this NADPH molecule can be utilized as a reducing equivalent, okay, as a source of electron. So tissues with active pentose phosphate pathway. So there are certain tissues where this pentose phosphate pathway is a little bit more active. Why? For example, steroid synthesis. So steroid synthesis again is an anabolic reaction, and this reaction requires NADPH, and this reaction mainly happens in the adrenal gland. So there is an active pentose phosphate pathway. So the liver is there. In liver, fatty acid synthesis, cholesterol synthesis happens. In testes, there is a steroid synthesis. In adipose tissue, there is a fatty acid synthesis. In ovary, there is a steroid synthesis. In mammary gland, there is a fatty acid synthesis. Red blood cell maintain the maintenance of the reduced glutathione. For this purpose, this pentose phosphate pathway are more active in the adrenal gland, liver, testes, adipose tissue, ovary, mammary gland, red blood cell. Okay, so these are the location where this pentose phosphate pathway is a more active. So this is the structure of NADPH. We call it as a reduced nicotinamide adenine dinucleotide phosphate. Okay, this is long form. Reduced, because hydrogen is present. That is why we are calling it as a reduced nicotinamide adenine dinucleotide phosphate. NADPH is a phosphorylated form of the NADP. Okay, it is a NADH. So we can observe the difference here. Here phosphate group is present, here it is absent. So this is a phosphorylated form of NADH. In general, with some exception, NADH is used to drive the phosphorylation of ADP to phosphate. So last lecture we saw under the Krebs cycle there is a production of NADH, and this NADH molecule can enter into the electron transport chain, and this electron transport chain facilitate the oxidative phosphorylation in which the ADP gets converted into the ATP molecule. So there is a role of this NADH, whereas this NADPH is used where reducing potential is required. Okay, so where this NADPH, so mainly its role is not like production of ATP. Main role is a reducing potential which is required in the synthetic reaction. In that case, this NADPH molecule is required. Okay, so this is a role of NADPH. So so we are saying the pentose pathway are divided into two phases. So there are two phases in the pentose phosphate pathway. We call it as the oxidative phases and the non-oxidative phases. Okay. So this phase is a oxidative phase, and here is a non-oxidative oxidative conversion of the interconversion of the sugar. So what happens is in oxidative phases, so there is some molecule are getting oxidized in these phases, in oxidative phases, whereas in non-oxidative phases there is just interconversion of the sugar is happening. So we have glucose 6 phosphate molecule here. This it get converted finally in the oxidative phase into the ribulose 5 phosphate. During this process 2 NADPH is generated. Okay, during oxidative phase, if somebody asks how many NADPH is generated, it is 2 NADPH is generated. Okay, then this ribulose 5 phosphate, it can get converted into this xylulose 5 phosphate or ribose 5 phosphate, and again ribose 5 phosphate and xylulose 5-phosphate combine, and we get glyceraldehyde 3-phosphate and the pseudoheptulose 7-phosphate. So this is a aldose sugar, ribose 5-phosphate. Xylulose 5-phosphate is a ketose sugar. They combine together, and we are getting this glyceraldehyde 3-phosphate and pseudoheptulose 7-phosphate. Okay. Again, what is happening is that during the non-oxidative phases, glyceraldehyde 3-phosphate and the pseudoheptulose 7-phosphate, they are combining, and we are getting fructose 6-phosphate and the erythrose 4-phosphate. So these two molecules we are getting. And at the next phase, erythrose 4-phosphate is combining with the different molecule here, xylulose 5-phosphate, and we are getting fructose 6-phosphate and glyceraldehyde 3-phosphate. In this way, we get fructose 6-phosphate, glyceraldehyde 3-phosphate, which are like these molecules are also present in the glycolysis, right? So common molecules. Okay. So here ultimately we are getting fructose 6-phosphate here. This is the pentose phosphate pathway. So NADPH and hydrogen ion present is formed from the two separate reaction. Okay. So how this NADPH is formed? So we are going in little bit detail. Glucose 6-phosphate dehydrogenase reaction is a rate limiting step and essentially irreversible. So first reaction, glucose 6-phosphate is there. Okay, so this is there, and now this glucose 6-phosphate, with the help of glucose 6-phosphate dehydrogenase, it get converted into 6-phosphogluconolactone. So whenever we say dehydrogenation reaction, this is a redox reaction. This NADP plus get converted into NADPH plus H plus ion, and enzyme required is a glucose 6-phosphate dehydrogenase. So this enzyme is required here, and at the next step, 6-phosphogluconolactone can get converted into 6-phosphogluconate, and the reaction enzyme required is the lactonase. Lactonase enzyme is required at the second step of the pentose phosphate pathway. And what is happening here is that the water molecule get incorporated, and we are getting additional hydrogen ion here. Okay. Next, 6-phosphogluconolactone gets converted into ribulose 5-phosphate, and the enzyme involved is a 6-phosphogluconate dehydrogenase, 6-phosphogluconate dehydrogenase, and the intermediates here are the NADP plus is getting converted into NADPH plus H plus, and here one carbon dioxide is getting removed. So we see pentose phosphate pathway is called as a pentose phosphate pathway. There is a 5-carbon sugar involved. This is a ribulose 5-phosphate. So what is happening here is that glucose 6-phosphate getting converted into 6-phosphogluconolactone. One NADPH is generating 6-phosphogluconolactone is getting converted into 6-phosphogluconate. Next, 6-phosphogluconate is getting converted into ribulose. So previously this was a 6-carbon sugar, 6-phosphogluconate. Now it has got converted into the ribulose 5-phosphate, and here is this 1 carbon extra carbon which got removed. So this is carbon is getting removed, and here also we are getting NADPH reducing equivalent, which will be required in the synthesis reaction. Okay, so so till now from glucose 6-phosphate to ribulose 5-phosphate, this reaction we call it as the oxidative reactions of the pentose phosphate pathway. So next, there are some isomerases, epimerases enzyme is there, which are causing the structural rearrangement. So if there is a phosphopentose isomerase enzyme is there, means this ribulose 5-phosphate get converted into ribose 5-phosphate. If there is a phosphopentose epimerase enzyme is there, then this ribulose 5-phosphate get converted into xylulose 5-phosphate. So we have two molecules, xylulose 5-phosphate now and ribose 5-phosphate, which are part of non-oxidative sugar interconversion. Okay, pentose sugar interconversion. Now these two molecules are there now, ribose 5-phosphate and the xylulose 5-phosphate, and there is a enzyme called as a transketolase. This transketolase enzyme make the structural rearrangement in these two sugar, and we get pseudoheptulose 7-phosphate and the glyceraldehyde 3-phosphate. So these two molecules we got. Next, with the help of transaldolase molecule, this pseudoheptulose 7-phosphate and the glyceraldehyde 3-phosphate they combine, and there is this again structural rearrangement. We are getting erythrose 4 phosphate and the fructose 6 phosphate. So here we got fructose 6 phosphate. This erythrose 4 phosphate can combine with this xylulose 5 phosphate under the transketolase reaction, and we can get additional molecule of the fructose 6 phosphate from the glyceraldehyde 3 phosphate. So we see this NADPH, which is producing this pentose phosphate pathway. It is required as a reducing equivalent for this synthesis reaction. And then this ribose 5 phosphate, which is produced here, it can be a part of like nucleotide synthesis, where deoxyribose sugar or ribose sugar, where this ribose backbone is involved. It can be supplied with the help of this pentose phosphate pathway. So again, same reaction. So we already saw this is oxidative pentose phosphate pathway. Glucose 6 phosphate is getting converted into 6 phosphogluconolactone, and the enzyme involved is a glucose 6 phosphate dehydrogenase. So dehydrogenase enzyme is there, means NADPH is producing in this step from the NADP plus. 6 phosphogluconolactone with the help of gluconolactonase get converted into 6 phosphogluconate. Here water molecule gets added and hydrogen gets removed, and at the next step 6 phosphogluconates can get converted into ribulose 5 phosphate. Ribulose 5 phosphate, and the enzyme involved is a 6 phosphogluconate dehydrogenase. So dehydrogenase is there, means NADPH is producing from the NADP plus, and carbon is getting removed. So here we see here is a dehydrogenase and here is a dehydrogenase. So whenever there is a dehydrogenase, there is a production of this NADPH here and NADPH here, which is a reducing equivalent, which we want to see in our pentose phosphate pathway. So ribulose 5 phosphate can undergo again the non-oxidative reactions. Okay, so this is a 5 carbon atom. So from the 6 carbon glucose, glucose 6 phosphate, we got 5 carbon sugar, ribulose 5 phosphate, and next non-oxidative reaction. So again in detail, if you see reaction structural arrangement and all those things, you can see here glucose 6 phosphate with the help of glucose 6 phosphate dehydrogenase is producing NADPH. So here one hydrogen atom is coming here, and one hydrogen atom which is present here and here is coming here. So that is why from NADP plus we are getting NADPH, and we are getting the sugar 6 phosphoglucono delta lactone, and with the lactonase enzyme water addition happens, and from the ring structure, so this is a ring structure, we get chain like structure. So this is like chain like 6 phosphogluconate, and 6 phosphogluconate dehydrogenase enzyme again gives the ribulose 5 phosphate. So here this carbon dioxide molecule end product gets removed here, and we get here a ribulose 5 phosphate. Glucose 6 phosphate with the help of glucose 6 phosphate dehydrogenase, which is a regulatory enzyme, this NADP plus gets converted into the NADPH, and here 6 phosphogluconolactone is produced. Okay, so basically this reaction only, first reaction, again we have shown as an individual reaction. So this enzyme is highly specific for NADP plus. The Km for NAD plus is 1000 times greater than NADP plus. So this glucose 6 phosphate dehydrogenase enzyme is very specific for this NADPH, means the probability is very high that NADPH is only get produced in this reaction. Next, 6 phosphogluconate delta lactone with the help of lactonase enzyme, the water molecule get added, and we get 6 phosphogluconate here. Okay, so 6 phosphogluconate. So this was a ring structure. Now it is a chain structure. Okay, 6 phosphogluconate. Now this chain structure with the help of 6 phosphogluconate dehydrogenase gets converted into ribulose 5 phosphate and the carbon dioxide. So here this already we saw this CO2 group get removed here, and we get ribulose 5 phosphate, and the enzyme involved is a 6 phosphogluconate dehydrogenase, and NADPH also generated in this process. So these are all the reactions of the oxidative pathway. Already we have saw glucose 6 phosphate with the NADP plus getting converted in 6 phosphogluconate delta lactone plus NADPH, and the enzyme involved is a glucose 6 phosphate dehydrogenase, then 6 phosphogluconate. This get converted into the 6 phosphogluconate. 6 phosphoglucono delta lactone get converted into 6 phosphogluconate with the lactonase enzyme, and this molecule again get converted into ribulose 5-phosphate, and carbon dioxide is produced, NADPH produced. So this is our NADPH we are getting, NADPH we are getting. Okay, and during non-oxidative phases, phosphopentose isomerase is there. So ribulose 5-phosphate get converted into the ribose 5-phosphate. So this sugar we are getting, ribulose 5-phosphate can get converted into the xylulose 5-phosphate with the phosphopentose epimerase, and then xylulose 5-phosphate and ribose 5-phosphate are combining. We are getting pseudoheptulose 7-phosphate plus glyceraldehyde 3-phosphate. Next, these reactions which is produced here, they again get combined here, and we get fructose 6-phosphate plus erythrose 4-phosphate. This erythrose 4-phosphate can combine with this xylulose 5-phosphate, and we can get fructose 6-phosphate and the glyceraldehyde 3-phosphate. Here transketolase reaction is involved. Okay. So these are the oxidative phases of the pentose phosphate pathway. So ribose can get converted into the ribose 5-phosphate can get converted into the xylulose 5-phosphate, and whenever ribose is getting ribose 5-phosphate is converting into xylulose 5-phosphate, the enzyme is a epimerase. So this is the enzyme we are showing here. Now what is happening there is that this ribose 5-phosphate and xylulose 5-phosphate, two different molecules, they can combine with the help of transketolase reaction, and we can get pseudoheptulose 7-phosphate and the glyceraldehyde 3-phosphate. Now we have got these two molecules. These two molecules can again combine, and the enzyme involved is a transaldolase. So pseudoheptulose 7-phosphate and the glyceraldehyde 3-phosphate, they are combining, and we are getting fructose 6-phosphate and the erythrose 4-phosphate. Now this fructose 6-phosphate can get interconverted into the glucose 6-phosphate. Erythrose 4-phosphate which is present can combine with the xylulose 5-phosphate. So xylulose was produced like this. So xylulose 5-phosphate, erythrose 4-phosphate and the xylulose 5-phosphate undergo the transketolase reaction, and we can get the glyceraldehyde 3-phosphate and the fructose 6-phosphate. Again, glyceraldehyde 3-phosphate, it can undergo the triose phosphate isomerase reaction, aldolase reaction, and the fructose 1,6-bisphosphatase reaction, and it can get converted into the fructose 6-phosphate, and this fructose 6-phosphate can again get converted into the glucose 6-phosphate. So basically these are the non-oxidative phases. Okay, here non-oxidative we call, because here we are not generating any NADPH or some oxidation is not there. Okay, this transketolase is involved here and here. This require thymine pyrophosphate. So everybody is knows that thymine is a vitamin, and this is required in our diet. So this is where this role of vitamins comes. Okay, so it acts as a coenzyme, thymine pyrophosphate, whereas this transaldolase reaction does not require this TPP, thymine pyrophosphate. The transketolase, thymine pyrophosphate, and the transaldolase are linked back to the glycolysis. Glyceraldehyde 3-phosphate and the fructose 6-phosphate are the part of this glycolysis. Basically, what is happening is that 3-carbon pentose sugar are combining, and we are getting 2,6-carbon sugar, that is fructose 6-phosphate. You can see here we are getting fructose, here we are getting fructose. This is 6-carbon, and we can get the 3-carbon sugar. Okay, this glyceraldehyde also we get here. Okay, so ingested ribose can enter the glycolytic pathway. Ribose can enter the glycolytic pathway through the pentose phosphate pathway. So 5-carbon sugar combining, 5-carbon sugar combining. Okay, and we are getting 7-carbon sugar, pseudoheptulose we were saying it. Okay, here only we are showing this reaction only we are showing on this slide. Only difference is that we are showing here the carbon atom. 5 combining, we are getting 7 and 3-carbon sugar. Then 7 3 combining, we are getting 6-carbon and the 4-carbon sugar. And now 4-carbon and 5-carbon combining, we are getting 6-carbon and 3-carbon sugar. And same reaction is happening here also. We are getting 6-carbon and the 3-carbon sugar here. Again, this 3-carbon can combine, we can get 6-carbon sugar here. So these are the non-oxidative phases. Already we saw this ribulose 5-phosphate can get converted with the epimerase, ribulose 5-phosphate epimerase into the xylulose 5-phosphate. If there is isomerase, the ribulose 5-phosphate isomerase convert this ribulose 5-phosphate to the ribose 5-phosphate. So xylulose 5-phosphate and the ribose 5-phosphate, they combine with the transketolase reaction, and we are getting pseudoheptulose 7-phosphate and the glyceraldehyde 3-phosphate. So this pseudohexylose 7 phosphate and the glyceraldehyde 3 phosphate, they are combining with the transaldolase reaction, and we are getting erythrose 4 phosphate and the fructose 6 phosphate. Erythrose 4 phosphate can combine with this hydrolose 5 phosphate with the transketolase reaction, and we are getting fructose 6 phosphate and glyceraldehyde 3 phosphate. Some multiple slides we have shown for the same thing, non-oxidative phases or the oxidative, to make you remember some names. Okay, so regulation of the pentose phosphate pathway. Glucose 6 phosphate dehydrogenase is the regulatory enzyme for this pentose phosphate pathway. NADPH is a potential competitive inhibitor of the enzyme. Okay, NADPH we are producing in the pentose phosphate pathway, and if already NADPH is present in the body, means it will act as a competitive inhibitor. It will say, okay, no need of NADPH, so it will try to inhibit this glucose 6 phosphate dehydrogenase enzyme. Usually the ratio of NADPH and the NADP plus is a high when enzyme is inhibited, but with the increased demand for NADPH, a ratio decreases. So whenever there is a demand for NADPH, this NADPH as the NADP plus ratio decreases, and enzyme, this glucose 6 phosphate dehydrogenase enzyme can get activated. Okay, so this is a regulatory enzyme. The reactions of the non-oxidative portion of the pentose phosphate pathway are readily reversible. So that is why we can see during non-oxidative phases there is a reversible arrow shown. The concentration of the product and reactant can shift depending on the metabolic need of the particular tissue. So product and the reactant, how much is needed, can be dependent on metabolic need. Okay, so if the cell is dividing very fast, at very fast dividing cell, we may require this NADPH reducing equivalent or some ribose as a component of DNA RNA. So based on that, this requirement may vary. Rapidly dividing cell require more ribose, so that is why we say rapidly dividing cell require more ribose 5 phosphate than NADPH. So we can see this ribulose 5 phosphate is a part of DNA RNA, so that is why it is required more. You can see glucose 6 phosphate get converted into fructose 6 phosphate, which can get converted into ribose 5 phosphate, or fructose 6 phosphate can get converted into fructose 1,6 bis phosphate. It again under the glycolysis can get converted into dihydroxyacetone phosphate and the glyceraldehyde 3 phosphate, and this glyceraldehyde 3 phosphate can again enter into the ribose 5 phosphate. So the need for NADPH and the ribose 5 phosphate is a balanced. You can see here glucose 6 phosphate is getting converted into the ribulose 5 phosphate, and during this process 2 NADPH can get generated and the carbon dioxide get removed. That is why this is a 6 carbon sugar, and carbon dioxide 1 carbon is removed. That is why this is a 5 carbon sugar, and this ribulose 5 phosphate can get converted into the ribose 5 phosphate. The more NADPH is needed, then NADPH is needed than ribose 5 phosphate in case of the fatty acid synthesis in the adipose tissue. Whenever fatty acid synthesis is going on in the adipose tissue, in that case, requirement of NADPH is more compared to the ribose 5 phosphate. So how this NADPH is generated? So this is the pathway. Glucose 6 phosphate is getting converted into ribulose 5 phosphate. Here we are generating 2 NADPH, and this is a non-oxidative. So ribose 5 phosphate is getting generated. It is getting converted into glyceraldehyde or the fructose 6 phosphate. So basically, in the fatty acid synthesis, we require more NADPH here. Okay. Sometimes cells needs both NADPH and the ATP. So here is the case. The similar reaction, glucose 6 phosphate getting converted into the ribulose 5 phosphate. Here we are getting NADPH here. NADPH we got here. Then this ribulose 5 phosphate can get converted into the ribose 5 phosphate, and then ribose 5 phosphate can get converted into the glyceraldehyde 3 phosphate, and the glyceraldehyde 3 phosphate by the glycolysis can get converted into the pyruvate. Here we are generating ATP. So in this case, when cell require both reducing potential in the form of NADPH and also needs energy ATP, so the cell resort to this pathway. So cell does this thing. It converts glucose 6 phosphate to ribulose 5 phosphate. It generates the NADPH, and then ribulose 5 phosphate get converted into ribose 5 phosphate, then glyceraldehyde 3 phosphate, and then the glyceraldehyde 3 phosphate undergo this glycolysis. So glycolysis pathway, and it generates the ATP for the cells. So that is how it adjusts, the cell adjusts itself, whether you require NADPH more in case of fatty acid synthesis or rapidly dividing cell where you need ribose 5-phosphate more, or sometimes there is a reducing equivalent NADPH is required more and as well as ATP is required more, then cell resort to different pathways. So this reaction, you can say this and this looks similar, but here glyceraldehyde 3-phosphate was there, but here this glyceraldehyde 3-phosphate got converted into the this pyruvate molecule. So cell adjusted. Okay, in this case I will need NADPH more, but in this reaction I need NADPH as well as ATP more. So that is why it resorted to this pathway. So glutathione and NADPH. So what is glutathione? Why it is important? How it is related to NADPH? These are the some questions you can ask about the glutathione and NADPH. So you can see glutathione is a tripeptide. So tripeptide means this is a peptide molecule. Peptide molecule contains amino acids. So how this glutathione is produced? Glutathione is composed of glutamate. This is a one amino acid. Cysteine is another amino acid, and glycine is another amino acid. So we have glutamate here in presence of ATP. This ATP get converted into ADP in inorganic phosphate, and during this process glutamate and cysteine combine together and we get gamma glutamyl cysteine. This gamma glutamyl cysteine combined with this glycine amino acid in the presence of ATP, where ATP is getting converted into ADP and inorganic phosphate, and now we have this glutathione. So what is present in the glutathione? There is a presence of glutamate, there is a presence of cysteine, there is a presence of glycine, 3 amino acid. So that is a glutathione, and this is a reduced glutathione. Reduced glutathione we show as a GSH. Glutathione function. So glutathione serves as a reductant. It conjugates to the drug, making them water soluble. So glutathione, because it is present as a GSH reduced form, it can act as a reductant. It will reduce other compounds or the substrate. It can conjugate with the drugs and making them water soluble. So some drugs are there which are not water soluble, and now we have this glutathione molecule in our body which will go and bind with those drugs and make them water soluble. It is involved in the amino acid transport across the cell membrane. So if you want some amino acid to transport across cell membrane, then this glutathione can help. So it can act as a cofactor in some enzymatic reaction like rearrangement of protein disulfide bond is there. Protein disulfide bond is there. If you want to rearrange those bonds, we want to break the bond between two methionine or two cysteine molecule, and we want to break and connect with the another cysteine or methionine. So in that case, this glutathione molecule is involved because it is acting as a reductant here. Glutathione functions, again sulfhydryl of GSH is used to reduce the peroxides. So this reactive oxygen species, so whenever there is oxidative damage, so there is a reactive oxygen species. So if you want to reduce the oxidative damage, in this case the sulfhydryl group, so this sulfur and this hydryl group of glutathione can use to reduce this reactive oxygen species formed during the oxygen transport. Okay, so for reducing this reactive oxygen species, this sulfhydryl group of the GSS is important. Reactive oxygen species damages the molecule. So what happens if you this reactive oxygen species remain unattended? Well, in that case, this reactive oxygen species can damage the macromolecules like DNA, RNA, and the protein, and they can ultimately lead to cell death. So whenever there is a more oxidative damage, in that case more and more reactive oxygen species is being produced, and if that is not got addressed with the help of reduced sulfhydryl group of glutathione, in that case cell death may happen. The resulting oxidized form of GSH. So but what is happening? The GSS, it is reducing this reactive oxygen species, but it itself is getting oxidized in that process, and this is resulting oxidized form of GSH is a two molecule linked by disulfide. So that is why we call it as a GSSG. So this is a two glutathione molecule with the sulfhydryl bond. They are linked together, and this oxidized GSSG is formed. The enzyme glutathione reductase uses the NADPH. So again, so what is function of NADPH? So we are talking about NADPH all the time. So this NADPH is function is that already from here we have got this GSSH. Okay, so this last one GSSH we got, GSSG we got, oxidized glutathione. Now we want to reduce it back to the GSH. Okay, in this case, the NADPH will say, okay, I will give this reducing equivalent to this one, and this GSHG goes back to the GSH. Thus, the pentose phosphate pathway is linked to the supply of adequate amount of the GSH. So we can see here the reduced glutathione, GSH, it will prevent the oxidative damage by reacting with this reactive oxygen species. But in this process, this reduced glutathione GSH get converted into this oxidized GSH, glutathione disulfide, and we want this glutathione disulfide in GSH form, reduced form, so that it will be again helpful whenever there is oxidative damage. So in that case, this NADPH which is produced via pentose phosphate pathway, it will come in action. It will say, okay, this is linked together. I will come here and I will break them and I will give one hydrogen here, and I will make GSH, glutathione, reduced glutathione from the oxidized glutathione. So again, this reduced glutathione will be helpful to prevent the oxidative damage. So what happens if the glucose 6 phosphate dehydrogenase is a defective, insufficient? So whenever glucose 6 phosphate dehydrogenase, which is the first enzyme of the pentose phosphate pathway, if that is a defective, then we will have less NADPH production. So there will look which translate into insufficient glutathione. So insufficient glutathione will be there. So is it a medical problem? So yes. So we already saw glutathione is important in the preventing oxidative damage and the preventing cell death. So it may have effect. So GSH is reduced GSH is extremely important, particularly in the highly oxidizing environment of red blood cell. So we see oxidative. Where are the chances of the oxidative damage? Is there more like reactive oxygen species? So which cells in our body carries the oxygen? So RBC is there. RBC, red blood cell, is the cell which have very oxidizing environment because the hemoglobin is attached with this oxygen molecule. So we are breathing in the air in our lung, and from the air we are taking in the oxygen. This is combining with the red blood cell, and then red blood cell are traveling at the tissue location to dislodge this oxygen at the required places and coming back again with the CO2. So basically this red blood cell is very they are present in very highly oxidizing environment. So here the role of GSH is important, reduce glutathione. Mature RBC have no mitochondria and are totally dependent on NADPH for pentose phosphate pathway to generate GSH from GSSG via glutathione reductase. So basically this RBC, they don't have mitochondria, so if they want a NADPH, they are totally dependent on NADPH for conversion of GSH from GSSG. Okay, in fact, as much as 10% of the glucose consumption by RBC is mediated by the pentose phosphate pathway. Okay, so 10% of the glucose consumption is mediated by this pentose phosphate pathway. The glutathione and the erythrocyte, reduced form of the glutathione, serve as a self-hydryl buffer. Okay, so reduced form is GSG, GSH. Okay, reduced glutathione, hydrogen is attached to here. So this reduced form of glutathione serves as a self-hydryl buffer, and it maintains the cysteine residue in the hemoglobin and other protein in the reduced state. So this cysteine residue present in hemoglobin can be maintained in the reduced state, or the other protein in the reduced state if you want to maintain. Then this sulfahydryl buffer of glutathione is important. GSH is essential for normal RBC structure and keeping the hemoglobin in the Fe++ state. So this reduced glutathione is needed here to keep this normal RBC structure, or the keeping the hemoglobin in the AP++ state. Again, reduced glutathione also detoxifies the peroxides. So we already saw the reactive oxygen species is there. Some peroxides are forming. So these peroxides are not good for the cells. They will go and bind with lipid. They will cause lipid peroxidation. The cell membrane get damaged. Then again more damage is happening. Cell is dying, which is not good for organism. In that case, this reduced glutathione 2-GSH is combining here with the ROOH here peroxides, and we are getting GSSG oxidized form plus water and this alcoholic form ROH. The cell with the low level of GSH are susceptible to hemolysis. So if the RBC have low GSH, it is high chance that this RBC will undergo the hemolysis. Individual with the reduced GSH are subject to hemolysis. Okay, so if your cells RBC don't have enough of reduced glutathione, then they may undergo the hemolysis. This is often clinically seen as a black urine. In this case, in this present, some black urine can be observed. The condition for hemolytic anemia related to glucose 6 phosphate dehydrogenase deficiency. Okay, ingestion of oxidative agents that generate the peroxides and the reactive oxygen species. Okay, so whenever there is oxidative reagents is there, it always tries to generate peroxides or reactive oxygen species. Okay, so in antimalarial primaquine or the purine glycosides from the fava beans, they can generate this antioxidants molecule, right? So individual with the glucose 6 phosphate deficiency, glucose 6 phosphate dehydrogenase deficiency cannot produce the sufficient reduced glutathione to cope up with the reactive oxygen species. So protein can become cross-linked, leading to the Haynes body formation and the cell lysis. So whenever this glucose 6 phosphate deficiency is there, you can see the protein are getting linked and this Haynes body formation is there, which is shown here. So this Haynes body formation can be observed in the cell. Glucose 6 phosphate deficiency and the non-spheroitic hemolytic anemia, over 300 genetic variation of glucose 6 phosphate deficient dehydrogenase protein are known. Thus, it is a remarkable variation in the clinical spectrum. Glucose 6 phosphate deficiency is inheritable X-linked disorder. So, how this disorder glucose 6 phosphate dehydrogenase deficiency transfer? So, it is a basically genetically linked, inheritable X-linked recessive disorder. Approximately 10% to 14% of the male in the African American population is affected by this glucose 6 phosphate dehydrogenase deficiency. It is also seen in Caucasians from the Mediterranean basin. Okay, the people with the disorder are not normally anemic, but and display no evidence of the disease until RBC gets oxidized. So whenever some people are there in which this glucose 6 phosphate dehydrogenase deficiency is there. If this deficiency is there, it means that NADPH will be lower. NADPH will be lower means that reduced glutathione will not get oxidized glutathione will not get converted into reduced glutathione. So there will be more oxidative damage. Chances are there they may suffer the hemolysis. So this drug that can precipitate this reaction. So people are there which are glucose 6 phosphate dehydrogenase deficient, and we are giving them antimalarial drug or sulfonamide or aspirin or non-steroidal anti-inflammatory drug or nitrofurantoin, quinine, or exposure to some chemical like mothball. So if you are, so these are some people and they are deficient with this glucose 6 phosphate dehydrogenase, and these are more prone to hemolysis, and in addition you are trying to give these additional drugs like antimalarial, sulfonamide, aspirin, then these people can suffer from this hemolysis reaction. So there is a favism where the individual with the glucose 6 phosphate dehydrogenase deficiency must not eat the fava beans. The erythrocytes can lyse and give the dark or the black urine. The growth of Plasmodium falciparum malaria parasite actually falls in the glucose 6 phosphate dehydrogenase deficiency. So this is one interesting finding. People have noted that if some people have glucose 6 phosphate dehydrogenase deficiency, in that case this Plasmodium falciparum, which is a causative agent of this malaria parasite, its growth got inhibited. Okay, so So that's it for today's lecture for the pentose phosphate pathway. Okay.